notes, our next unit, a little subunit, will be on translation. That's going from our DNA code that was brought out by the mRNA into our actual protein. And it's very important that that happens correctly because it's the order that those bases are laid down. There's A's and T's and G's and C's, the order those bases are laid down in the DNA has to be copied correctly in the mRNA. And then for the mRNA, they have to be translated correctly and picking up the correct amino acids, those 20 amino acids that are floating around dissolved in that cytoplasm. They have to be laid down in the correct order for proteins to function correctly. But sometimes the order gets messed up. Something happens. It may be during transcription, or it may be in translation. Sometimes something happens and a mutation occurs. So we're going to talk a little bit about mutations as well in this little segment here, in this lesson. So the nature of the genetic code. Ribosomes make proteins by stringing amino acids together to form long chains called polypeptides. And we talked a little bit about that before, how amino acids are the building blocks of proteins. You get an amino acid, maybe you might code for a valine, which is one of the amino acids, and that valine might be stuck to a serine. And then a serine might be stuck to another valine. And then these bonds between these are called peptide bonds. Those are peptide bonds that form between those amino acids. And those peptide bonds, where you get a really long chain, your poly means many, you get a really long chain of these valines and these serines and these 20 amino acids that are listed here, with maybe an arginine or a histidine. And you get a big old long chain of those. It's called a polypeptide because it has many peptide bonds. So it would be a polypeptide. Well, big old long polypeptide, that's what becomes a protein. So ribosomes are made by make proteins by stringing together these amino acids. There's only 20 of them, but it's the combination, the order they go in, that determines what the protein is and what it does. Okay. So amino acids are linked together by these peptide bonds. Each polypeptide contains a combination of any or all of the 20 different amino acids. Okay. And it's important that we get those amino acids in our diet. We may get them from eating proteins, and then our body breaks down the proteins, so those amino acids are floating around in our cytoplasm. Some of them are essential. We have to eat them. Some of them, our bodies can make some, and our bodies cannot make others. So those will be essential ones that is important to eat. So DNA and RNA contain 4 bases, and these bases are arranged in groups of 3. Remember, it's those groups of 3 that we called triplets earlier. Those groups of 3, each set of 3 bases would code for 1 amino acid. So from valine, if we look at valine right here, one that might code for valine would be, we have a guanine and then a uracil and then a uracil. Well, that would code for a valine. Or if we have a uracil and then a cytosine and then an adenine, that would code for a serine amino acid. Code word for DNA bases is copied into a strand of mRNA through the process of transcription. Remember, transcription happened first. It was in the nucleus because it's DNA being copied into a complementary strand of mRNA. So transcription occurs in the nucleus every time because the DNA cannot leave the nucleus. It has to happen in the nucleus. So each combination of the 3 bases of mRNA is called a codon. Codon. You ever got a little decoding chart in it? Maybe you had something that was written in code. Sometimes it's shapes, sometimes it's letters. It would give you a code and say, oh, and really this stands for that. So you can decode something. Well, that's kind of what the codons are. You have this set of maybe A-U-G, and the A-U-G is going to code for something. Or the C-C and C, and it's going to code for something. So you have to be able to decode it. The ribosomes have to be able to decode it or translate it to turn it into a protein. And those are codons. It's a code that is written in. So AUG that I just mentioned, AUG is important because it serves as the starter. It's AUG, that's the starter. And it tells the ribosome, okay, right here, this is the starting place. This is where you start making this protein. And I remember it because AUG is abbreviation for August, and school usually starts in August. School starts in August. AUG is our starter codon. It starts all of our proteins. So if we look at our chart, how it works is AUG, A, our first letter's on this side, our second letter's there, our third letter's there. So we go A, we're going to be on this row. The U, well, that tells us we're going to be in this box right here. And then our third is G, so methionine. Methionine right there is our starter amino acid. That's the amino acid that's going to start each protein. A, U, G. All right, we're going to use the wheel. We start in the center and work our way out. There's A to U to G. Methionine, right there. And there are three stop codons. UGA, and I remember because UGA can always stop Alabama. So UGA, UAG, and UAA. Those are all going to signal stop. Okay? They're all going to signal stop. So if we go to U, to G, to A, that tells us it's a stop. Or we can do it this way. We can go U, be on this one, to A, so that one, and then G, and there's our stop right there. So that would tell the ribosome, stop, you're finished with the protein, it's time to stop. Those are our stop codons. It's the end of that polypeptide. So the three main types of RNA, we briefly spoke about this earlier. So mRNA, messenger, carries that message from the DNA in the nucleus out to the cytoplasm. tRNA transfer, or what I like to call tow truck, because I picture it as it has to put on a special hook for each amino acid, and it goes, hooks it up and brings it back to the ribosome. So tRNA transfer or tow truck RNA transports the amino acid dissolved in the cytoplasm back to the ribosome to be stuck together and assembled into a protein. So the correct translation of the mRNA depends on the joining of these correctly in the order from the mRNA codon, the correct tRNA molecule, which is called the anticodon. The hook, that tow truck's going to put on for a specific amino acid, that would be our anticodon. It just means it's anti-opposite of the codon that was on the mRNA. And then we have rRNA. R, ribosomal RNA, or I like to call it R for reader RNA, because it's going to proofread. The ribosomal RNA is going to read those instructions. It's going to proofread. It's going to read those instructions. It tells it what order to put those amino acids in, and it's going to proofread and make sure they're in the correct order. So it clamps onto the mRNA and uses its information to assemble those amino acids into the correct order. That's the rRNA. And this process is called translation. The process of decoding an mRNA message into a protein. So mRNA to protein. Translation translates mRNA to a protein. So if it's translating an mRNA into a protein, where in the cell is it happening? If I say protein, you say ribosome. It's going to happen in the ribosome because that's where proteins are made. Ribosome. Translation. mRNA translated into a protein. Happens in the ribosome. So the genetic code is a code that translates the DNA language into a protein language. So the genetic code goes from DNA to protein, translates that DNA language into protein language. And once that message has been translated, the protein can be made. So translation to protein. So Francis Crick came up with this idea that's called the central dogma. It's kind of an odd name. But Francis Crick, Watson and Crick, is a little older now. Back in 1953, discovered the double helix of the DNA. Well, later on, he's going to call it the central dogma. And the central dogma just means we go from a DNA to an RNA and from RNA to protein. DNA to RNA, RNA to protein. Francis Crick called that the central dogma. That's how it happens. So mutations. Sometimes in all of this copying of stuff, things get kind of out of order, or a base might get stuck on incorrectly, and we end up with what's called a mutation. So they occur when the base is A.T.G.C joined incorrectly. So joining incorrectly results in a change called a mutation. Okay, think about Teenage Mutant Ninja Turtles. Things in they were exposed to the chemicals that made things get out of whack, the bases misaligned, and they end up with mutations. Okay. So something that causes a mutation would be called a mutagen, as in Teenage Mutant Ninja Turtles. They're exposed to mutagen. Okay, so mutations. A wrong base in a DNA, so maybe the DNA was copied, something happened, and there was a miscopy when a cell was replicating, and maybe a wrong base ended up in the DNA, and then that would mess up everything from there on out. So if there's a wrong base in the DNA, it's going to give the cell the wrong message, and the result will be the wrong type of protein. And it's all going to go back to that DNA code. If there's a base that's wrong here, then something's going to end up wrong with it. Sometimes it can be dramatic, and sometimes it's just a little thing that happens that doesn't even change anything. So other than copy mistakes, how else can mutations occur? Okay, sometimes maybe there should have been sometimes maybe it should have been an A, a G, a C, another C. Well, it may end up with an A and somehow ends up with a C and a C and a C. That would be, just got misplaced. That would be a substitution. Sometimes that G may get knocked out altogether. You may end up with ACC. That would be a deletion. Sometimes it might get carried away and it might really like that G, and it might somehow end up sticking that down a few extra G's. And that would be insertions or replications. So different things can happen as you're putting those bases in there that can cause misprints or mutations. Any agent that can cause a change in DNA is called a mutagen. So some examples are chemicals such as drugs and alcohol that can cause mutations. Radiation such as X-rays or even UV rays from the sun. UV rays from the sun cause genetic changes in skin cells that can lead to skin cancers. That's a mutation caused by the mutagen of UV rays, radiation from the sun, and from tanning beds. Okay, so quiz time. DNA to RNA to X. What would that X be? DNA to RNA to protein. So the genetic flow above represents the cellular process of transcription and translation. What word best replaces that X? That would be protein. And that's Crick's central dogma. Our amino acids link together to form proteins. Okay, so we may have a valine connected to a cytosine, connected to a histidine, connected to a glycine, connected to a glycine, and they're all held together by peptide bonds. Peptide bonds. Peptide bonds form a big, long polypeptide. That polypeptide will be a protein. So which RNA is responsible for carrying the code from the nucleus? Carrying it is carrying the message. So it is a messenger RNA, mRNA, messenger RNA. Carries that message from the DNA in the nucleus out to the ribosome. Which codon serves as the starter for protein synthesis? Remember we talked about school starts in August. So AUG is our start codon, A.T.G.C. Start codon. This thing's not working. Okay, mutation is best described as change in the DNA sequence. If there's a change in the DNA sequence, that would be a mutation. answers if you